Good morning, everyone. This is Kalavati Ramachandran here, and as a part of our group project, I'll be talking a little bit about sequence ontology basics and why why do we have to use genome annotation library. So, GAL is basically a sequence ontology tool developed by the sequence ontology community for working with the biomedical and sequence ontology data. So, what is sequence ontology? Like we have seen during our class modules, sequence ontology is a set of terms and relationships used to describe the feature and attributes of a biological sequence. It includes different kind of features which are located on the genome sequence. As you can see in this ontology tree, several features like gene, transcript, mRNA are all part of the sequence features in sequence ontology, and they all have a special kind of relationship. For example, gene is a sequence feature, and transcript is part of a gene, and mRNA is a transcript, and so on. So before I go on to each and every sequence feature, just a quick note on Sequence Ontology Project. It is a collaborative project which was originally established by the Gene Ontology Consortium. It's a large user community developed for the definition of the sequence feature used in the biological sequence annotation. Next is the gene sequence feature, which can be defined as a region that includes all of the sequence elements necessary to encode a functional transcript. A gene may include regulatory regions, transcribed regions, and or other functional sequence regions. This is sort of a table, a parent and a children table. Gene group and biological region are the parent for the gene sequence feature, and on the right hand side you can see the list of children that fall under the gene sequence feature. Next is transcript, which can be defined as an RNA synthesized on a DNA or RNA template by RNA polymerase, and this has a parent as gene member region, and its transcript children transcript are listed on the right. This is basically ontology of the transcript. Here on the right hand side we can see mature transcript, primary and process transcript is a relationship, while transcript region is part of a transcript. Next on the sequence feature is mRNA, which is a messenger RNA sequence. It's a mature transcript sequence, a portion of which is coding. It may include UTR, the untranslated region, but not intron sequence. As we can see in this diagram, mRNA is follows a relationship with process transcript, and process transcript is a transcript. Intron is part of a primary transcript, and primary transcript derives from process transcript. However, intron is not an mRNA. It is just part of the primary transcript. And this is the parent and child table for the mRNA, a mature transcript being the parent, and on the right it has a children tree. So this kind of gives the sequence ontology for the same. Attenuator mRNA and riboswitch are the part of mRNA, polyase adjacent to the mRNA, and EST derives from mRNA. So these are special kind of sequence ontology relationship that have been described here. Next is sequence alterations. Sequence alteration is a sequence feature whose extent is a deviation from another sequence. It is merged with partially characterized change in nucleotide sequence. Parent being sequence feature, and its children are listed as on the table here, and can be clearly seen that all the children are is a relationship with sequence alteration feature. So we saw the basics of the sequence ontology and the relationship for the genome features. Now, why do we need to use genome annotation library? What's the benefit of GAL? And so GAL is a scripting language as presented by Joseph. It uses Perl programming language, which is a very powerful yet easy to write and easy to develop. It suits well for the bioinformatics or biological data, which are often incomplete, and it has irregular expression matching and string manipulation operators for interconverting incompatible data. So that's a key, and that's one of the prime reasons GAL is quite easy and straightforward. GAL uses a powerful sequence ontology relationship to obtain relevant and useful data from genome annotated sequence. And it provides numerous parser classes to convert other format to GFF3 or GBF. Ali will be talking more about the parser and parser classes, and that's about it. Thanks for your time.